Hello everyone, welcome back to Igcse.science.notesz, and in this video we'll be completing inshallah the whole chapter of cells from the international AQA A level bio revision videos, the playlist thing. So we've done already biological molecules in 2 videos. Please go check them if you would like to register this chapter. And we've done the first part of cells already. In this video we'll be completing inshallah the second part. I believe this is very useful for international AQA, somehow in Excel and OCR. As we said, we've done already biological membranes in the last video. In this video we'll be starting with the movement of the molecules. And I want you to know that there are types of movement of molecules, and it depends on the properties of the molecule, which type will we be using and the requirement of the cell. So, starting with the diffusion, we all know that it's the passive movement from high concentration to an area of low concentration, and the molecules move directly through the phospholipid layer. Also, you need to know that it's movement of small non-polar lipid-soluble molecules, such as carbon dioxide and oxygen. The facilitated diffusion requires a channel protein, and so it transports polar molecules, charged and water-soluble molecules across the membrane. The osmosis is the diffusion of water molecules from an area of high water potential to an area of low water potential, a partially permeable membrane. Active transport is basically active, not passive as diffusion and the osmosis. The active transport, active. Why is it active? Because it requires energy in the form of ATP, and it transports all types of molecules through a carrier protein from an area of low concentration to an area of high concentration. The exocytosis and endocytosis transport large particles. The particles are enclosed in vesicles made from the cell surface membrane and transported into the cell in endocytosis. In exocytosis, vesicles contain large particles and are fused with the cell surface membrane and released from the cell. The co-transport uses ions to move substances into and out of the cells. This occurs particularly in the epithelial cells of the ileum. Here, sodium ions and potassium ions are pumped out of the epithelial cell by active transport into the blood, leaving a lower concentration in the cell, which causes these ions to move in from the lumen by facilitated diffusion, which at the same time brings glucose and amino acids into the cell. These then diffuse from a high concentration in the epithelial cell to low concentration in the blood, and the rate of gas exchange by diffusion becomes more rapid as the surface area of the surface increase, the diffusion distance decrease, the diffusion gradient becomes more steep, and the temperature increase. Okay, let's get deep into more other components of more other organisms of the life. Bacteria and viruses. Bacteria and viruses are the main disease-causing pathogens in humans. Even though they both cause disease, they vary in many ways. Their differences are as following. Basically, the bacteria are prokaryotic cells, and their genetic information is stored in the form of circular strand of DNA, whereas the virus consists of just nucleic acid enclosed in a protein coat, and their genetic material can take the form of DNA or RNA. The bacteria do not require a host to survive, whereas the viruses are entirely independent on their host and cannot survive without them. The viruses are significantly smaller than the bacteria, and bacteria have a cell membrane, cell wall, and cytoplasm, as well as other organelles such as ribosomes, plasmids, flagellum, and pili, whereas virus possess no such structures. That's completely amazing. Okay, what is an example of bacterial disease? An example of bacterial disease is a tuberculosis, also known as the TB. The TB is caused by bacteria called Mycobacterium tuberculosis, which infects the phagocytes in the lungs. The first infection is symptomless, as the infected phagocytes are sealed in tubercles as a result of inflammatory response in the lungs. However, the bacteria lie dormant inside the tubercles as they are not destroyed by the immune system due to the tubercles being covered with a thick waxy coat. When the immune system becomes weakened, the bacteria becomes active again and slowly destroy the lung tissue, thus leading to breathing problems, coughing, weight loss, as well as fever. The TB can potentially lead to death. An example of a viral infection is the HIV. I think all of you heard about it. The human immunodeficiency virus, which causes AIDS. The first symptom of HIV include fever, tiredness, and headaches. After several weeks, the HIV antibodies appear in blood, thus making a person HIV positive. After this period, the symptoms disappear until the immune system becomes weakened again, thus leading to AIDS. Symptoms of AIDS include weight loss, diarrhea, dementia, cancer, and opportunist infections such as the ATP. Okay, What about our immune response? There are physical barriers and chemical barriers. The physical barriers to infection include the skin, the stomach acid, and the gut and skin flora. The skin, which is a tough physical barrier consisting of keratin, stomach acid, which kills bacteria, gut and skin flora, natural bacterial flora, competes with pathogen for food and space. The immune system responds to antigens which are found on the surface of cells, and these are proteins that have a complex structure that is unique for each cell and therefore identifies the cell as self or non-self. What is the non-specific immune response? The non-specific immune response of the body to infection include the inflammation, the lysozyme action, the interferon, the phagocytosis, and here's a diagram of it. Okay, so the inflammation is basically the histamines released by damaged white tissues cause vasodilation, which increases as the blood flow of the blood to the infected area and increases the permeability of blood vessels. And as a result of that, antibodies, white blood cells, and plasma leak out into the infected tissue and destroy the pathogen. The lysozyme action are enzymes. The lysozymes are enzymes found in secretions such as tears and mucus, which kill bacteria cells by damaging their cell walls. The interferon prevent viruses spreading to uninfected cells by stopping protein synthesis in viruses. The phagocytosis is a process in which white blood cells engulf pathogens to destroy them. They do this by fusing to a pathogen and enclose them in a phagocytic vacuole with a lysosome, and see the diagram below for detailed process. After the pathogen is engulfed and destroyed, its chemical marker called antigen are then presented on the surface of the phagocyte, and the phagocyte that becomes an antigen presenting cell, which activates an immune response if the antigen is recognized as a foreign. As you see over here, the phagocyte is attracted to a pathogen. Receptors go in, digest, bind, done. Okay. What about the specific immune response? The specific immune response is antigen specific and produces responses specific to one type of pathogen only. This type of immune response relies on the lymphocytes produced on the bone in the bone marrow, the B cells and the C cells. The B cells mature in the bone marrow and are involved in the humoral response. The T cells move from the bone marrow to the thymus gland where they mature and are involved in the cell-mediated response. The cell-specific immune response glossary: memory cells, B effector on or plasma, T helper, T killer, and the memory cells are cells which replicate themselves, which exposed to an invading pathogen and remain in the lymph nodes for decades researching for the same antigen. This results in a much faster immune response result in a much faster immune response and should the individual be affected by the same pathogen again. The B effector or plasma cells are antibody producing cells. The T helper cells stimulate B cells and T killer cells to divide. The T killer cells destroy pathogen infected cells. Here the cell mediated response. T cells directly destroy the antigens, and the helper T cells stimulate the T and B cells. The suppressor T cell inhibits the T and B. The memory T cells remember antigen for future encounters. Also here at the beginning, the foreign microbe with the antigen, macrophage ingests antigen, present them to the T cell then produce the clones. Okay, here's the humoral response, where A B cells is triggered when it encounters its matching antigen. The B cell engulfs the antigen and digested, then it displays antigen fragments bound to the unique MC cells. This combination of antigen and MHC attracts the help of a mature matching T cells. Then cytokines secreted by the T cell help the B cell to multiply and mature into antibody producing the plasma cells and release into the blood. Antibodies lock onto matching antigens. The antigen antibody complexes are then cleared by the complement cascade or by the liver and spleen. The antibodies in the humoral response plasma cells, they produce antibodies, and these are made of 4 polypeptide chains forming a Y-shaped structure, as you see in the right. These are complementary to only a single antigen, and they work by forming an antigen-antibody complex, which serve as markers for phagocytes to destroy the attached cells. Due to antibodies having 2 binding sites, they can also clump cells together, making them easier for phagocytes to find. This process is called the agglutination. Okay, so the antigen binding site, light chain, heavy chain, receptor binding site, variable region, different antibodies, and the constant region. Now we go to the immunity part. The immunity can either be active or passive. The active immunity results from the production of antibodies by the system in response to the presence of an antigen, whereas the passive immunity results from the introduction of antibodies from another person or animal. These are also two subtypes of immunity, natural or artificial. The natural active immunity arises from being exposed to an antigen and getting the disease, whereas the natural passive immunity is the result of crossing of mother's antibodies through the placenta and their presence in the breast milk. The active artificial immunity is acquired through vaccination, which stimulate the immune system and lead to production of antibodies, whereas the passive artificial immunity is where antibodies are injected into the body. The vaccines are a way of introducing a pathogen into the body in order to produce an immune response. The pathogen may be dead or inactivated, but the antigen on its surface will still produce an immune response. This is an example of active immunity and results in the creation of memory B cells, which will be able to trigger a rapid secondary immune response should the same pathogen ever be detected again. Vaccines is different than sorry, I messed something up. Vaccines is an active artificial immunity, whereas being exposed is the natural active immunity. Okay. So let's complete. The success of a vaccination program is dependent on a number of factors such as the cost of the vaccine, severity of the side effects, ease of production, transportation and administration, and number of people who need to be vaccinated for herd immunity. The concept of herd immunity is that if you vaccinated enough people in the population, then eventually the pathogen won't be able to be transmitted from different hosts. This therefore means that those who aren't vaccinated are protected by those around them who are. And I'm going to be explaining it in further details when we solve, inshallah. However, vaccines are not always useful in preventing a disease outbreak. This is because the antigen on the surface of the pathogen can change, therefore removing immunity. An example of a virus that does this is influenza. This virus rapidly changes its antigen, and therefore immunity against the virus is only short-lived. Ethical concentration. There are a number of ethical considerations to take into account when looking at vaccines, and these include the production and testing of vaccines may be done on animals. The risk of the vaccine need to be balanced with the benefits. The vaccines must be tested on humans first to determine toxicity. Vaccinations are very expensive, and should vaccination be compulsory, or should people be able to opt out of having a vaccination? Be optional. Okay. The monoclonal antibodies. Due to there being many different types of antigens, each B cell will produce a different complementary antibody, and therefore medical useful to be able to produce many clones of a single type of antibody. These are known as the monoclonal antibodies, and they have many different uses as seen below. Direct therapy, which basically the monoclonal antibody that are specific antigens found on the surface of cancer cells can be used to target and then destroy the cell as part of an immune response. For example, the Herceptin targets breast cancer cells. Or by the indirect therapy, which drugs can be attached to monoclonal antibodies, such as the cytotoxic drug. Then the antibody then is used to direct the drug towards the cell, displaying a particular antigen rather than towards other the cells. And the diagnosis, a particular antigen are targeted by antibodies to measure levels of that antigen in the body. Lastly, the pregnancy testing. Monoclonal antibodies in home pregnancy kits are specific to the hormone human chorionic gonadotropin. Human chorionic gonadotropin. Okay, and then we go to the HIV, which we've talked about earlier in simple details. Now, we need to know that HIV, the pathogen, can lead to disease-acquired immune deficiency syndrome, which is basically AIDS, and HIV has the following structure: lipid envelope with embedded attachment proteins inside the protein capsid, where the genetic material, which is the RNA and reverse transcriptase enzymes, are present. This catalyzes the production of DNA from the RNA, and in order to replicate, the HIV virus binds to the protein CD4, which is most frequently bound on T helper cells. Capsid then fuses with the cell surface membrane and the RNA, and reverse transcriptase enter the cell. The reverse transcriptase converts the DNA to RNA, which then moves into the nucleus of the cell. The cell now has the instructions to begin in producing viral HIV components. To identify the HIV, an enzyme-linked immune sorbent assay, ELISA, can be used. This detects the presence of quantity of the antigen found on the HIV, and ELISA is shown in the diagram below. Antigen coated well, wash, add specific antibody to be measured, wash, add enzyme conjugated secondary antibody, wash, add substrate, and measure the color. Antibiotics are effective against HIV and viruses. This is because many antibiotics work by preventing bacteria from making cell walls. Without this, the bacterial cell cannot control the entry and exit of water and will therefore burst. However, since viruses do not have a cell wall and are produced within a host cell, they are unaffected by antibiotics. Instead, HIV is treated with antiretroviral drugs which keep the levels of HIV in the bloodstream very low, reducing the impacts on the host's immune system. Thank you so much for watching this video. I'll be very glad if you leave a comment telling me if I need to change something, if it was very useful for you or whatsoever. Leave a comment, please. Have a nice, beautiful day. And now we're currently done with the first 2 chapters, finishing the 3rd chapter in the very soon time, so you'll be able to revise all the chapters as soon before your exam. Have a nice day and Ramadan Kareem.